Hi guys, so we're covering chapter 10 today, talking through effects of landscapes on community structure and dynamics. So communities are made up of species that interact to varying degrees within the same geography. We know communities are shaped by a variety of factors, both abiotic and biotic, climate, the level of productivity, disturbance history, colonization and extinction dynamics, evolutionary processes, like we talked about last week, human land use history, and species introductions are all going to play a role. So there are two ways to really study landscape effects on community ecology, and both are important. The bottom-up approach to understand individual populations usually misses the more complex interactions among species. We know that communities are more than the sum of their parts. However, it is important to be able to really fully understand individual populations that exist within a community. And then a top-down approach would examine how landscapes structure influences the properties of communities, as well as searching for general patterns in the distribution of diversity. And ultimately, both the bottom-up and top-down approaches are needed really to fully understand dynamics of a community and its interacting parts. There are a few metrics that we see used in understanding community dynamics, and specifically diversity of species. Species richness is the number of species present in a given location. This measure is obviously sensitive to sampling effort. So this can be graphically displayed with a species accumulation curve that compares sampling effort with the species total number. If we're thinking about sampling at a site or patch and comparing that to other sites or patches, it's of course important for us to standardize that sampling effort. Species diversity is a function both of species richness and relative abundance. The most commonly used index is the Shannon Diversity Index. And it's important to remember both for richness and diversity that these are indices and not actual measures of biodiversity. So just as the radius is an index, the volume of a circle, these are measures that are indices as well. The refraction curves that we discuss, there's a great example in the book here that it talks about a study that was conducted with arthropods. Arthropods were sampled in an experimental landscape that varied in the amount of habitat fragmentation. Only 10% of the habitat cells were sampled, and so sampling effort was much greater in the landscapes having more habitat. And that obviously could lead to a bias in understanding habitat area effects. In order to correct for this, the curves were calculated in the figure below by repeatedly resampling the individuals collected and plotting the average number of species expected for a given sample size. So in the end landscape, with a different number of species, gain species still at approximately the same rate. There are a few different sort of spatial ways we can look at diversity indices, and the book explains this in some depth. Alpha diversity is the number of species within a given location. Could be a sampling location or a small patch. It is equivalent to the local diversity, while gamma diversity is a measure of of total species across a larger landscape or region, and it's often applied to a larger collection of samples. Beta diversity is the degree to which species composition differs or changes among communities. This is a figure that gives a little insight into that way we can look at assessing beta diversity. There was another interesting study that was discussed in the book that assesses community similarity along a spatial or environmental gradient. We understand that, that there can often be a shift, and we have tools to help us look at communities along a gradient. In this case we were looking at aquatic communities, stream assemblages, and looking at distances and how those communities turned over. So in all three groups of taxa that were studied, we saw a similar response to distance along the stream gradient. So they all exhibited a significant turnover in community, somewhere around 23 to 54 kilometers. Another study looked at community assemblages associated with places where beavers had been extirpated, forested wetlands, forested riparian areas, in comparison to beaver sort of generated habitats of meadows and shrubby swamps. And what's really interesting here is that in the shrubby swamps and meadows that were both being affected by the engineering of beavers, we saw similar overlap in community type, and then almost no overlap in that place where beavers had been removed. So just a nice visual representation of the role that beavers play as ecosystem engineers, and the uniqueness of the communities that follow along with that. So throughout the semester, we've revisited a few different times this space-time diagrams that help us conceptualize how various factors work into the hierarchy of landscape ecology. And in this case, we're looking at the different ways we can look at species richness, first within communities or habitats at that smallest scale, but scaling that all the way up to landscapes, geographies within continents, and then richness across continents and up to the global level. And so at that broadest scale, the study of biogeography is the study of species diversity patterns at broad scales. And one thing we do now know is that scale matters when understanding patterns of species diversity. So we, in particular, can talk about latitudinal gradients that we see. We know that that latitudinal gradient of diversity is one that there are many different hypotheses surrounding. The general understanding is that diversity increases as we approach the equator. We can see this in the figure of global hotspots for diversity. And the productivity hypothesis states that great productivity results in a greater number of species. If we think of the trophic levels of species in an ecosystem as a pyramid, that wider base of primary producers that we might see in the tropics would of course lead to greater number of species as we move up those trophic levels all the way to the top. And of course, primary producers are ultimately limited by climatic factors. Another hypothesis, the climate stability hypothesis, postulates that the relative evolutionary stability of the tropics has led to greater diversification there. So while some northern latitudes have seen numerous glaciation events, of course, the tropics have been spared those over time, just leading to a longer, stable time for diversification of species. So that's resulted in the tropics having species with narrower ecological niches. This niche conservatism concept may also explain why so many tropical species show the flexibility or generality in order to make latitudinal shifts and range expansions. And so this theory also relates to the cradle of life hypothesis, which just presumes that more species originated in the tropics than in other places, partly because of that stability. A few of the other theories that the book talks about is the habitat heterogeneity hypothesis, museums of biodiversity, the out of the tropics hypothesis, and then the Red Queen hypothesis. So the Red Queen hypothesis I decided to pull out because we've touched on brood parasitism before, but it's a great example of an evolutionary arms race that we see between parasites, in this case brood parasites, and their hosts. And what we see is that over evolutionary time, brood generalists, like the brown-headed cowbird, would eventually evolve into host-specific parasites because the interaction between host and parasites, they're constantly having to one-up each other in this evolutionary arms race, which would eventually lead the brood parasites to having to specialize on a single host and mimic their eggs exactly. And that's what we've seen with some of the cuckoos in the Old World. And you can see that in the graphic below that we have these single species parasites who mimic their eggs exactly. So we've talked about now the diversity that results from latitudinal differences. Let's talk a little bit about elevation gradients. So we know that mountains cover about 25% of the Earth's surface. That creates a lot of different climatic conditions in a localized area. So both elevation and slope aspect contribute to the diversity that we see. You might remember that it was the study of elevation, zonation, and plant communities that first led to the study of landscapes through a German geographer by the name of Karl Troll. That gave way to the life zones concept that we discussed earlier in the semester as well. And this relationship between species diversity and elevation is somewhat complicated by latitude, not fully straightforward. So we see somewhat different trends in different latitudinal areas. One of the things we have seen is that elevation diversity can, in some cases, be hump-shaped rather than linear. So we saw that in a plant study in the diversity of species in the Pyrenees. And that kind of fits with some botanists' conclusion that there's a mid-elevation area that's ideal for many plant species, leading to plant diversity peaking at mid-elevation and being slightly lower in lower elevations and then really dropping off as we get to the highest elevations. So we see here 4 different patterns of elevation diversity. You can see that generally the pattern is that as elevation increases, diversity decreases, but we see different trends with that, including that that mid-peak trim, or the slight hump trend in that yellow line. And what you'll see is that most plants, as well as non-flying mammals, show the highest diversity in that final mid-peak elevation shape, whereas birds, interestingly, sort of are evenly distributed among these different patterns. Let's talk a little bit about species area relationships. We've certainly visited this at length during this semester, but it is such an important principle in landscape ecology. I don't think it hurts to return to this. So we know that species area relationships are arguably the most ubiquitous scaling rule in ecology. In its simplest form, species increases as a power of area. But there's more than one type of species area relationship, and we have different sampling designs that might give rise to different types of relationships as well. So this figure shows a variety of curves that might result from different sampling techniques. One, the nested sampling design. Two, a continuous or non-continuous sampling design, and three, the island or patch-based sampling designs. So these design types ultimately influence which type of diversity we are measuring. And a nested design might not lead to a measure of alpha diversity, for instance. So species area relationships are often extrapolated across scales to predict the relationship between species richness and habitat area. But we know that diversity can be scale dependent and will not always exhibit the same slope. So we need to be careful when we're trying to extrapolate a diversity measure. The book revisits, again, something you all are well versed in now, the island biogeography theory and habitat fragmentation paradigm. And so, you know, we know that MacArthur and Wilson, from when they first wrote their paper, anticipated the application of the theory of island biogeography to fragmented landscapes. And really these island biogeography principles have dominated the study of fragmented habitats ever since. So especially in the way we design fragmentation experiments, the relationship between patch size and isolation, however, do not always follow the exact principles of the island biogeography theory. So the ultimate result, we now know, can depend on a few factors. So what type of taxonomic group is being studied, the composition and the relative abundance of species in that community, and even specific traits of those species. So we now have a better understanding of characteristics of species that are going to be most affected by patch area. We know, of course, it's somewhat intuitive that species with large body size are going to be impacted by the amount of space available to them. So larger species generally require larger areas to meet their energetic needs. They tend to occur at lower densities, and for that reason are always going to be sensitive to area. And then we also know that rarity in general, whether it's because they're specialist or species that require larger areas, rarity inherently increases the risk of extinction from a patch. For example, the slope of the species area relationship for rare species were at least 3 times larger than those for more common species, and that was shown across 3 different trophic groups. And specialists are intuitively exhibit also higher sensitivity to patch area than generalists, as well as top consumers. And then sedentary or alternately very mobile species as well. There was an interesting study I wanted to highlight where the researchers investigated the effect of patch size reduction on the composition and the functional richness of avian communities. They focused on some morphological traits associated with seed dispersers in frugivorous birds. And they found that there were three key traits that were important. One was the hand-wing index, which is essentially a measure of body size, body mass, and the gape width. That's the actual width of the bird is able to open their beak, and is a signal of what size fruit they can ingest. And so decreased, all of these decreased. These three factors decreased with patch size reduction. And because species with larger values for morphological traits were lost through local extinctions. So the relative absence of large gaped and more dispersed frugivores and small forest fragments has important implications because these species play a pivotal role in seed dispersal, carrying a lot of seeds, a high number of seeds, and also consuming the larger size seeds that the smaller birds would not be able to disperse. So there's an intuitive impact here on plant communities if we lose some of these larger seed dispersers as patch area becomes smaller. It's easy to see that there's somewhat of a confounded relationship between traits that impact a population and their sensitivity to area, so the size of the patch, versus their sensitivity to isolation effects, the distance between patches. So rarity, specialization, and trophic position all contribute both to patch isolation effects significantly. Notably, the difference is the impact of body size. We see more impacts related to area on large-bodied animals and more of an impact on isolation with some of our small body size. And that, of course, relates to them being poor dispersers generally. This figure considers the relative impact of both area and isolation on a variety of species-specific traits. So it can be difficult to determine if patch area or isolation has a more significant effect on diversity. As we've said, the two are often confounded, but there is general consensus that area is relatively more important than level of isolation. So we've discussed habitat loss and fragmentation are confounded and linked. To some extent, researchers have been able to disentangle these two effects experimentally. So in review, the loss of habitat amount is typically found to have large negative effects on biodiversity, while the effects of fragmentation tend to be weaker. Habitat amount hypothesis states that species richness is predicted to be related to the amount of habitat rather than configuration. So it really creates a test to disentangle these two differences. So this depends on us being able to define landscape at a scale appropriate for the taxa that we're studying. And this approach has a lot in common with patch connectivity and nearest neighbor measures that we've talked about before. So here, sampling locations rather than the patch itself are going to be the measure. Predictions of habitat amount hypothesis. Here are a few of the tenets. So holding patch size constant, the number of species at a sampling location should increase as the amount of habitat in the landscape increases. Site-based richness should not vary among patches in different sizes if the overall amount of habitat in the landscape is the same. And then species richness at a given site should increase with increasing habitat amount in the surrounding landscape. even as the size of the patch decreases. So, as you might imagine, studies like the 2016 plant study have refuted this patch area hypothesis, demonstrated that local patch size actually has a profound and significant effect on plant diversity. So this, again, is a type of null hypothesis that we might prop up to test against to better understand the effect of configuration. And a concluding premise is that spatial configuration does actually matter. So the amount of habitat loss, the isolation of habitat, or conversely its connectivity, the matrix quality, all of these things we know can influence patch scale attributes such as the patch area, the patch shape, and the complexity, including edge effects. And so arrows in this figure show some of the causal inferences, which are going to vary depending on the species or community of interest. So the community response to these factors depends on the species response as well. And so what we don't have shown here in the response is actually those interactions among the species in the community. So we've talked about niche theory already, and of course when we think about communities, we revisit that, the biological role of species played in an environment. We talked in previous lectures about competitive exclusion and the concept of competitive release, and then also about behavioral differences that we might see, what we would call interference versus exploitation, competition, all to say the role of competition among species in a community is very important in determining the species diversity there and some of the patterns that we see in interactions with the landscape. We've briefly visited this study and the concept of resource partitioning, but I wanted to bring this up again. So Robert MacArthur, again, was the source of the classic study on foraging ecology and wood warblers that looked at and demonstrated resource partitioning in these small woodland warblers that basically concentrated their foraging efforts in various parts of the tree and differentiated in where they specialized in foraging in the tree. This really helped us understand a little more about interspecific competition and how communities structure themselves. Another important concept is predator-prey dynamics. In the absence of predators, we would expect prey to distribute themselves where resources are most abundant. However, as density increases with high-quality resources, so will inter- and intra-specific competition. And we can anticipate that predators are going to respond to a density in prey. So, in a sense, quality should also be assessed in accordance with predation risk. And this has important influences on community structure. So predators are more likely to match the distribution of their prey when spatial overlap is assessed at broader scales. There may be a little overlap between predators and prey when their distribution is assessed at more fine scales. Some predator-prey studies assume a homogeneous landscape. In the field, we often don't actually see this perfect relationship as we see in this graph, which is a representation of the classic Lotka-Volterra predator-prey model. So some other studies have led us to the conclusion that spatial heterogeneity in the presence of spatial refugia actually has a stabilizing effect on predator-prey interactions in areas where predators would otherwise overexploit their prey. And so in that case, landscape structure is very important and part of the interplay in this relationship between predators and prey. And generally, we can think of predators and prey as engaging in a behavioral space. Predators prefer areas where their prey are most abundant, whereas prey are going to avoid those areas. And the degree of spatial overlap between predators and prey influences the strength and the stability of their interactions and their likelihood of coexistence. And we discussed early in the lecture that predators are more sensitive to habitat loss and fragmentation, and especially so if they are specialists. However, generalist predators may actually benefit from an increasing matrix or more disturbance in a landscape that provides them with more prey resources. The book goes into parasite-prey or parasite-host interactions as well, and we see many of those same nuances that we see in predator-prey dynamics. Because it's so important, it's worth pausing and thinking about habitat loss and fragmentation and its effects on pollinators. We know that loss, fragmentation, and degradation of natural and even semi-natural habitats has contributed to global declines in bees and in other pollinators. This habitat loss and fragmentation can affect pollinators in at least 3 different ways. It alters plant density. It alters the density of the pollinators themselves that often are social beings, and to some extent their structure of their communities depend on a certain critical mass. It alters the foraging and dispersal behavior of pollinators over time. So we know habitat loss has a more profound effect on plant and pollinator densities, while the fragmentation is really going to impact pollinator movement patterns. And pollination services are critically dependent on having a sufficient amount of habitat within the foraging and dispersal range of the pollinators that they depend on. So in this case, corridors between patches may actually help to promote the movement of pollinators and increase pollen flow. And finally, the book goes into some detail about metapopulation structure and dynamics, 4 frameworks that we might test in landscape ecology. The patch dynamics perspective assumes that the habitat is patchy but homogeneous and that species differ in dispersal abilities. A species sorting perspective might assume a heterogeneous landscape in which all species can disperse freely but can coexist regionally because of different niche requirements. And then the mass effects perspective uses source-sink dynamics to explain regional coexistence. Species differ in flex rates due to source-sink dynamics. The neutral model perspective assumes that species are ecologically equivalent, don't exhibit niche differences or differences in dispersal, speciation and immigration rates, offset extinction, which are driven by drift rather than competition or exclusion. So this model really serves as it's a null hypothesis framework to help us understand what patterns would look like in the absence of some of the processes that we know are at play. And that's it for chapter 10.